Hello, everyone. Welcome to Brainwaves. I'm Brandon Staglin. So, as I like to say often, cribbing a page from Aristotle's playbook, we humans are social animals. Our relationships sustain us and make us stronger. However, in certain conditions like schizophrenia or autism, people's brain wiring may develop in unhealthy patterns, leading to them to socially withdraw at times. And this may impair their ability to feel fulfilled in their lives and function well in life. So if we can help these people find opportunities to improve their social function, it may help them to improve their mental health in general and their overall. So with us today is Dr. Dev Manoli, the newest IMRO Assistant Professor of Psychiatry at UCSF. And he's doing some really interesting research to better understand why social function might go awry in diseases like schizophrenia and autism, and how attachments form, and in order to better understand how to improve the social and mental health of our population. So I'm going to interview Dev, and you'll be able to ask him questions either live by this Google Plus Hangout or after the Hangout on inhro.org brain-waves. Dev, thanks so much for being with us today on Brainwaves.
Thanks, Brandon, very, very much for the invitation.
You're very welcome. So I'm going to jump right in with questions. Okay. So yeah, I know your research on social function has focused primarily on animal models like prairie voles, for example, these little rodents. How can studying the relationships function of prairie voles and other animals improve our understanding of how human relationships and the ability to develop relationships develops?
So I think that's a really, really important question in terms of why are animal models so important to really understand the neural basis of mental illness. And there are a couple of reasons why I think just in general, and I'll start at the most general levels, why animal models are important. And the first thing is because in a model system, you can really control for everything from the genetics of the organism to the specific manipulations that you want to do. And that's really important to sort of understand the cause and effect relationships between whether they're genetic mutations or whether they're environmental factors that really contribute to mental illness. So that's why we study model systems in general, to really understand and in an experimental way test our hypotheses. And then obviously the utility of an animal model is that you can go ahead and actually make the manipulations or alter the genes of the animal or different parts of its neural function, which obviously in clinical populations is something that we don't want to be doing unless we know that there is a direct benefit to a patient. So that's kind of just the general bird's eye view. And the specific reason for prairie voles is, so they're these really fascinating rodents that form lifelong attachments, and we call those pair bonds. So unlike many other mammals, and in fact about 96% or 95% of mammals don't form adult long-term relationships, these animals sort of bond for life. Once they've mated, they have this pair bond that really persists. And so now we have a really good model for adult attachment behaviors and really able to look at how the brain actually encodes attachment at a neurobiological level. And so then the question in terms of how this really impacts our ability to understand human dynamics is really because of the fact that as far as we can see so far, the mechanisms that prairie voles use and other animals, as well as humans, to really form these attachments seem to be conserved. And so we really focus a lot of our work on two hormones, one of which is called oxytocin, another one is related to it called vasopressin. And in prairie voles, they're really principally responsible for the ability to form a bond between mates. And it turns out that in humans, they really are critical players in human attachment behaviors too. So while there's no single way to really explain how they work, we know that both oxytocin and vasopressin really modulate trust-related behaviors, affiliative behavior, or our ability to form relationships in humans too. So again, now we can really start to understand how do these things work, and then certainly in the context of different mental illnesses, what goes wrong.
That's really cool stuff, and it makes sense. So, based on what you've learned from your research in voles, from a neurobiological standpoint, why are deep, resilient social relationships so important for people's mental health?
So, I think it basically goes back to the original quote that you brought up from Aristotle, in that we are social animals. But then the question is, what does that actually mean? And actually, a lot of work done by people long before I started work on Prairie Voles really has shown that as a consequence of the ability to form a pair bond, there are a lot of other behavioral correlates that really are also changed in Prairie Voles, and we also know in people. So for example, when animals mate and form a long-term relationship, that facilitates the care of the young. So like people, Prairie Voles, both males and females, really show high levels of parental behavior. Another aspect of that is that when you separate, for example, when we are separated from our partners or our loved ones, we feel distress and we feel anxiety. And there are correlates of those sorts of behaviors in Prairie Voles too. And so the fact that we are social animals has really sort of led to, from a neurobiological standpoint, to mean that our brains are really built to facilitate these interactions. And that becomes really important, in particular, when we think about treatment, and we can definitely talk about that more. But that it also promotes the overall health of the animal. And you can think about that from an initial standpoint, certainly from the care of the young and things like that. But when you get to larger animals or sort of more complex social structures, then you have the ability for animals to learn from each other. And so one of the thoughts might be that the ability to form social structures or social relationships really sort of forms the basis for the cultural transmission of information in species that really actually are able to live long and pass on information between the generations. And then the last point is also just that the ability, because our brains are really built even from childhood, whether you're a social species or not, to really interact with other individuals, as we know, pups will attach and children will attach to their parents, that those early experiences really promote overall health. And so as a consequence, unfortunately, in conditions where deprivation or abuse can occur, we know that that has very, very long-term and very dramatic, widespread consequences for the overall health of a person or a prairie vole. And so that's another, going back to your original question, there's another very important reason to really study these neural circuits, to really understand how early experience translates through adolescent development into changes in adult behavior as well.
Okay. Yeah, that makes a lot of sense too, and it jives with my experience. Well, I haven't mentioned this yet, but I don't even know this. I have schizophrenia, and when I was unable to see people on a regular basis because I felt isolated, I was isolated. I didn't feel I could interact with people. I found I needed a reality check every so often, and it's talking with people helped me to get that, So that leads me to my next question, and that is that in diseases like schizophrenia, like I have, in my case, when I was very symptomatic, I found it very difficult to make friends and even appreciate the love of my family at times. And what is happening in the brain, as you found from your research, when these challenges with social interaction develop for people?
So I think that's a really important point. And the short answer is right now, we don't know. But we do know that a couple of different things are happening. And one of the other important points that I think is also sort of a correlate, and I'll get back to the specific question you asked, is that you mentioned that the experience of people with schizophrenia in many cases can involve difficulty with social interactions. The other point is also, though, that the experiences of people with schizophrenia are not the same for everybody that has it. And so the question as to why one person's experience with schizophrenia is very, very different from another person, even though to a certain extent we do call them both schizophrenia and there are a lot of shared experiences, has a lot to do potentially not only with the environment in which they have developed in, but also the specific genetic background that they have. One person with schizophrenia has different genes that might be affected than another person. And so then the question is, how do individual genes really contribute to differences in the way people experience that? And so, the term that we use clinically is something called an endophenotype. And that basically just means this person or this population with schizophrenia has a certain set of symptoms versus this population, and how do individual genes contribute to one set of symptoms versus another. And so that's another reason why, for example, in Prairie Voles, we can now individually establish what's the cause and effect relationship between, for example, a specific mutation. But what that gets to is really sort of understanding what's happening in the brain in general, which is what you asked for. And so what we know from sort of clinical studies is that not only do people often with schizophrenia often have difficulty sort of experiencing emotions in the same way, but we know now through a lot of sort of clinical neuroscience research that they actually perceive things in a very different way as well, at the level of emotional stimulus. So for example, how they perceive faces, how they perceive just general social interactions is different. And that doesn't mean that it's not there, but it's just different. And how it's different is what we really want to get at. So why is it, for example, that somebody with, for example, with autism may feel frightened when they make direct eye contact in some cases, but still find social interactions with their parents or with other children very rewarding when they occur in a certain way? We don't know. And so the question is, how is the brain changed that these sort of sensory stimuli that they experience are encoded a little bit differently? The other thing we also know is that the thresholds, for example, for emotions are different for people with autism or people with schizophrenia, just as two examples. And so one of the terms that we talk about is emotional expressivity. And in the same way that someone with schizophrenia often can experience joy and sadness and anger in all of the same ways, if they experience it in their environment from other people, the intensity can be very, very overwhelming at times, even though it's the exact same emotion, because their biological thresholds are a little bit different. And we don't understand the mechanisms behind that at all, but we do know that it's important, for example, when a young person is starting to deal with some symptoms, that it's important to regulate emotions in their environment, because otherwise they can actually experience them as a physiologic stress.
Mm-hmm, mm-hmm. Wow, oh, Dev, there's some echo. Did you unplug your microphone or anything? Or
no, actually.
Okay. I don't hear it now.
Okay.
Now I hear it. I hear some echo. Hopefully that won't cause an issue with the broadcast. But anyway, we'll proceed. So research, to learn more about how the brain might change as social function changes, and because then you can maybe figure out how to treat it with biological or pharmacological or genetic treatments, and that leads me to my next question. One thing that improved my ability to socialize when I was recovering from schizophrenia, well, there are a few things, many things, but two that stand out for me were cognitive training, which improved my ability to process auditory stimuli coming in, maybe kind of like what you were talking about a moment ago. I believe that it helped there. And also more recently helped me to be calm and interact with people in a calmer way. What are in your view, some of the best treatments to improve social function, be they psychosocial or pharmacological or digital or any other way?
So I think there's actually a lot of really exciting research in sort of what are the best ways to really improve social cognitive function. And not only are they're motivated not only just by the importance of just social function in general, but also because a lot of earlier studies actually had shown that one of the most important prognostic factors for example, for people with schizophrenia is their baseline social function even before they develop other psychotic symptoms, for example. And so what we realized is that it's a very good indicator for sort of overall mental health and mental function. And really exciting research that's coming out from a number of different groups. For example, work with Carrie Bearden, Sofia Vinogradov, who's here at UCSF, is actually showing that when you use sort of social cognitive training, analogous to the sort of auditory processing training that you were talking about, that not only do you improve a person's ability to experience or interpret emotions and respond to them in a way that isn't, for example, through anxiety or fear responses, which we know that certain patient populations have a very strong predisposition for, but that when you actually start to use this sort of training, you actually activate parts of the brain that generalize the ability for the brain to sort of learn new processes. And that has to do with part of the reason why we find social experiences fulfilling in the first place, that we are motivated to be social. It activates the parts of our brains that make us feel good. And the nice thing is that actually also increases our ability to learn and function in other domains as well. And it's very interesting because at least what the preliminary research seems to be showing is that it's not necessarily true the other way, that even when we sort of exercise our executive function and improve our working memory or learning, although there are certain cases in which it is true that it does generalize that way, that those parts of our brain seem to be much, much more responsive when we're socially trained, even though if we leave social training out of it, that doesn't improve on its own, even if we improve sort of, for example, executive function. And so I think there's a lot of really interesting work from a behavioral standpoint, and like you mentioned, the cognitive training in terms of new therapy interventions for patients. From the pharmacologic side, I think that that's a really important point of why animal models are so important. Because right now, we know that in certain people, there's a significant benefit when you start to, for example, treat someone with oxytocin. But that's not the case for everyone. And we don't understand why. And we also know that something like oxytocin, which has a lot of effects, really, really can have benefits and negative consequences even in the same person. And so that way, I think it's really important to understand the basic neurobiology so we can get better sort of pharmacologic treatments to really, really specifically treat different sets of symptoms and symptoms in different patient populations.
That's awesome. You can, I mean, I didn't know the detail about how social cognitive training generalizes to improve executive function and other cognitive functions, and how social interaction is so important for learning. But that does make a lot of sense. So, thanks for explaining that. So, what research projects, and you mentioned some of it already, what research projects toward improving social function are you most excited about, and why?
So I think the social cognitive training and really coupling that with some of the cognitive neuroscience to really understand what are the parts of the brain in humans that are activated when we socially interact with other people, what are the parts of the brain that sort of motivate us to be social, and also that in patient populations aren't necessarily activated in the same way, and how do we actually start to co-opt different strategies or different mechanisms to really engage those circuits to sort of improve people's lives and their function. But then also, certainly I'm a little bit biased when I say this, really understanding how do we sort of dissect this out at the really neural circuit level, molecularly, to really understand how can we develop very, very specific interventions, either behaviorally or pharmacologically earlier, long before someone maybe has all of the symptoms, for example, of schizophrenia. And so that's actually one of the things that I'm, at least from our own work, really, really excited about, is really starting to understand what's happening in these circuits during development and adolescence that transforms early patterns of behavior into adult patterns of behavior, but also gives us a little bit of an insight into the biology so that we can start to intervene earlier, detect first before we intervene, and more specifically, so that we have better treatments that we can use to sort of supplement the behavioral interventions as well.
Yeah, that's really cool stuff. With the early intervention, that is critical for improving people's chances to live fulfilling lives and to recover from the early symptoms that they have. I observed in my own case too. I got pretty early intervention myself, helpful for me. So thanks for explaining all this, Dev. Incidentally, the Bearden Lab just posted a comment for you on the Hangout.
Oh, great.
It said hi, Dev. Thanks for the shout out.
Oh, of course. Great to hear from them. And, and as I mentioned, they're doing some really amazing stuff, especially as you mentioned, in early intervention and detection. And, I think that's the sort of work that actually motivated me to pursue the lines of research that we're pursuing.
Cool. Well, great. So, Dev, if people have any, after this broadcast is over, are you ready to answer some questions there for them?
I would love to. Please, I encourage people just to ask or make any comments that they have. But yeah, I look forward to answering questions.
Oh wait, that's great. But Bearden Lab just posted a question for you on this Hangout. So it says, as you know, there's a huge amount of heterogeneity in outcomes of nasal oxytocin trials in humans, both in healthy individuals, those with ASD and schizophrenia. Clearly, there are a lot of reasons for this, but based. So that it seems to end there. They might post a follow, like the second part of that in a moment.
Okay.
Just we can wait for that discussion. I learned some things I didn't know, and that's one of my favorite parts about doing these interviews is things that I learned from it, and getting to interact with one amazing scientist like you. All right, here's the second part of the question. Clearly, there are a lot of reasons for that. I'll just start over and read the whole thing. As you know, there's a huge amount of heterogeneity in outcomes of nasal oxytocin trials in humans, both in healthy individuals, those with ASD and schizophrenia. Clearly, there are, based on your basic science work, what do you think is the direction this work should go in?
So, I mean, I think that is that the point that they made, that the heterogeneity is a really important feature. And I think that really understanding the interaction between the genetics underlying different populations in autism spectrum disorder or in schizophrenia, and whether or not that actually specifically can predict the sorts of responses to an intervention like oxytocin. And so you can imagine that different genes operate in different parts of the brain in different ways. And so, a person with, for example, one set of mutations might have oxytocin be beneficial because it actually sort of engages the circuitry that's just a little bit not as easily primed to respond to social interactions, versus someone else with a different set of mutations might actually have some of the negative effects. So we know that oxytocin or vasopressin in some cases can actually inspire anxiety type behaviors. And so really, I think understanding the correlation between responses in human populations and the underlying genetics, so really starting to be able to genotype large groups of people that have different responses and different sort of baseline sets of symptoms, and then obviously translating that into the animal models and saying, okay, if I make mutations in X, Y, Z genes versus A, B, and C, and then I give an animal oxytocin or vasopressin, and I see differences in the response, can I start to understand why and where in the brain that's happening? Thanks very much for that question. It was a great feed.
That's great. That makes a lot of sense too. And I hope people at the Stanley Center for Psychiatric Research and the Psychiatric Genomics Consortium are listening to this, is what you just said as well, because that's a great insight too. So any other? I don't see any other questions coming in. So great. So thank you so much, Deb, for your amazing insights and all the research you do. And I look forward to talking with you again soon.
I do too. Thank you very much for the invitation, and thanks very much for the support that you guys are providing, because we wouldn't be able to do it without that. So I want to also acknowledge that.
Very welcome, and very happy to do that soon.